Hello, everybody. Welcome back to the Chestnut Week at the Pennsylvania Farm Show. I'm Jim Searing, president of the Pennsylvania and New Jersey chapter of the American Chestnut Foundation. I'm hosting this series, which is designed to inform and update you on the quest to save the American chestnut and restore it to our homes, our farms, our fields, and our forests. We're featuring an array of experts on growing, restoring, and finding chestnuts in our region. And we're really glad you joined us today. You know, we're here at the virtual Pennsylvania Farm Show, and that screen behind me is not in Harrisburg, it's in my home, but the screen behind me is the display we bring to Harrisburg every year, where we normally engage with thousands of people. We look forward to next year when hopefully we can see you all in person and online like we're doing now. You know, before we start our program, there are a few things you need to know. First, this program is being recorded. That includes anything that's entered into questions and answers or the chat. Secondly, we will mute everyone except for the presenters. And we've set this Zoom event up so that no one can see your screen. Lastly, we will not be surprising anyone by calling on individuals. We will have time for questions after Bill's presentation. So if you have one, please enter it in the Q&A box at the bottom of your screen. There's also a chat section, but we will not be monitoring it for questions. That's what the Q&A box is for. And if we don't have time to get to your questions, we'll try to come back to you with an email response. And incidentally, I just want to say one of the pleasures of this week, it's been really fun when our audience uses the chat box to let us know where you're from. We want to issue a welcome to all our members, members from other TACF state chapters, and all of you who are interested in our quest. So, and with that, let's get into our program. The American chestnut was one of the most important trees in the forests of eastern North America, from Mississippi to Maine and into Ontario. Foreshadowing all the destruction of trees that we're seeing today, the chestnut was almost entirely wiped out of those forests, starting in 1904, when a blight from Asia was first discovered to be killing trees in the Bronx Zoo. Just as we have a pandemic today affecting humans, chestnut faced its own tree-demic with devastating consequences. At the time, there were no vaccines and no solutions. By the 1950s, the United States had lost 3 or 4 billion chestnut trees, who's counting, and the species is considered critically endangered and functionally extinct today. The American Chestnut Foundation, or TACF for short, was formed in the 1980s to save the tree and find a way to restore what was lost. Today, the effort to save the American chestnut is considered one of the most ambitious ecological restoration programs in the world. Dr. Bill Powell is a director of the American Chestnut Research and Restoration Program at the State University of New York's College of Environmental Science and Forestry, or ESF, which is much easier to say. Almost 3 decades ago, Dr. Powell and Dr. Charles Maynard were asked by citizens to take a path less traveled, and one that was really in its infancy at the time, to see if biotechnology could somehow be used to save the American chestnut. For many years, breeding American trees with Asian chestnuts, which had resistance, was the focal point to develop a blight-tolerant tree. Over time, our organization came to understand that we needed multiple approaches because one method by itself was unlikely to solve the problem. After 35 years of research, discovery, and dogged determination under Dr. Powell's leadership, with public support, ESF has developed a blight-tolerant transgenic tree that's restricted to research orchards. Today, I'm really happy to say there's a 290-page petition. An incredible amount of work went into this, but there's a 290-page petition in front of the USDA laying out all the research and extensive safety testing on what's called the Darling 58 American Chestnut. And hopefully, after it has gone through an exhaustive regulatory review, there will come a day when we can all plant trees that are much more tolerant of the blight and able to sustain themselves in the wild and in our backyards. With that, welcome Bill to Chestnut Week at the PA Farm Show.
Right. Thank you, Jim. Can I go ahead and share my screen now?
Go right ahead.
Okay. Let me just share this real quick. Okay, well, thank you for this opportunity to come and present our research at the Pennsylvania Farm Show. So what we are part of this 3-burr collaboration with the American Chestnut Foundation. 3-burr stands for Breeding, Biotechnology, and Biocontrol United for Restoration. And so what I'm going to present today is our part of this, which is mainly the biotechnology side. So to start with, though, I can never, wait a minute here. Already technical difficulties. There we go. I always have to start with a little bit of background. I know you had a talk earlier in the week giving you a lot of background on American chestnut, but I just want to make sure everybody's up to snuff with why the American chestnut is important. This is a scene from the Blue Ridge Parkway in North Carolina. And about 100 years ago, if you were to look at that scene, about 1 out of every 4 trees would have been an American chestnut. This is one of the few pictures of an actual chestnut forest. This is the Shenandoah National Park in 1912. And if you look at that white on top of this trees there, what you're seeing are the actual male flowers, the catkins of the American chestnut. And people used to describe this as almost like snow in June when they saw the flowers all come out and bloom. It had many different values. It had a valuable wood, which was straight grained and rot resistant. It had a very consistent nut crop. You could eat the nuts raw, roast it. You can grind the flour. You can even brew them into beer. You know, it actually had medical uses too. The Native Americans used the leaves and the bark to treat certain ailments. Today, actually, people are studying extracts from European chestnut leaves to fight staph infections. And of course, they're really part of our culture. You can't go to a town or a city without seeing a chestnut street, just like you see in Maple Street or Oak Street. There was a time in the fall in the past where people used to go out chestnutting. And of course, we just got through the holiday season where I'm sure you heard the song that begins chestnuts roasting the open fire. That is actually talking about the American chestnut. Of course, at our school, the College of Environmental Science and Forestry, one of the things that we're most interested in is its impact on the environment. And I like to say that chestnut supported everything from bees to bears. Once it was lost, it had a devastating effect on wildlife. And of course, we don't have all those values today because of a introduced blight called the chestnut blight. So what happened? Well, a little over a century ago, people started importing Asian chestnut trees. And at the time, they didn't realize when you import a tree, you're not just importing that tree, but you're importing all the microbes that are on that tree. And it turns out there was one fungus called Cryphonectria parasitica that came over with the Asian species. And this fungus had co-evolved with these Asian species, so it didn't cause much damage on them. But our American species were very naive to this fungus and had no defenses. So this fungus would jump off the Asian species onto the American chestnut. It would colonize a wound in the chestnut. Once it colonizes that wound, it starts changing its physiology. It starts forming what's called mycelial fans, which are kind of mechanical wedges to help pry its way into tissues. And ahead of those fans, it produces acids, the main one being oxalic acid, as well as enzymes that then eat the dead tissue and supply the fungus with food. This forms a structure called a canker, shown in this picture, which eventually girdles a tree and cuts off all circulation and killing the top of the tree. Now, of course, when the blight came through, people were devastated. They couldn't, understand what was going on. As Jim mentioned, it took only about 50 years to go through the chestnut range from Maine down to Georgia and killing over 3 billion of the tallest trees in the forests. Now, because of that, people weren't just sitting still. People were trying all kinds of things to eradicate the chestnut blight and to bring back the chestnut tree. Obviously, at this point, they couldn't prevent it, but there was things like trying to test fungicides. They did mutational breeding, silviculture practices, use of hypervirulence, species replacement. All these things have been tried and have not succeeded. Today, you actually can go out and buy chestnut trees still, but most of what you buy today are either the Asian species or the chestnut hybrids. And just wanted to let you know right up front that there's nothing wrong with chestnut hybrids, but they are really for certain purposes. For example, they're great ornamental trees, and they are also very good for crops. But if you think about restoration, going back into a wild forest, they're really not adapted to do that. And that's because the species of chestnut evolved in different parts of the world, and they're adapted to these different environments. And when you start making hybrids of them or moving them around, they're not going to have all the characteristics needed for their original ecosystem. So hybrids have actually been produced since the 1920s. There's papers all the way back there describing them. Probably there were hybrids actually made before that. And today you can buy hybrids actually at the Walmart. One of the most popular ones is the Dunstan chestnut, which is a nice hybrid chestnut tree. And this just shows some of the nuts from that. But again, this would be great for agriculture, maybe for certain wildlife, but for restoration, it's not really the best tree. And that's because the American chestnuts actually produce a smaller nut. And why is that important? Well, it gives more opportunities to more wildlife to actually benefit from the chestnut tree. For example, this picture, actually a picture from TACF, one of their contests, showing mallard ducks eating chestnuts. And you can see that those mallard ducks probably couldn't choke down those large chestnuts shown above them, but they could do a regular American chestnut. Also, it turns out that the susceptible chestnut species, the American chestnut and the European chestnut, are the only canopy trees, the really tall trees of the chestnut family. The Asian chestnut trees have been actually bred for probably thousands of years to be more of an orchard tree. And so they're much smaller. And when you make a hybrid between those, you're going to get something that's intermediate between them. Now, it's important that the chestnut tree can compete in our forest and reach the canopy, because to flower, they need direct sunlight. And if you can't break the canopy, they're not going to flower and reproduce. Also, hybrids, a lot of people don't realize this, but hybrids, you're doing a lot to the genome of the chestnut tree when you're making a hybrid. The chromosomes don't exactly match, and you're mixing genes, again, from species that evolved in different parts of the world. So a lot of times you'll get things like reduced growth or intermediate traits, sometimes loss of the desired traits in future generations. Male sterility is a really common thing with hybrid chestnut trees. There's been reports of a condition called cracked bark, as well as internal kernel breakdown. Internal kernel breakdowns where about 40% of the nuts actually just kind of disintegrate inside their shells. But most of these things can be managed and can also be bred out in future generations. So again, there's nothing wrong with hybrids. It's just that I want you to know that there could be possible problems with it. And I'm going to make a comparison to what we do with genetic engineering to kind of show that you can avoid a lot of these problems using the biotechnology techniques. So to do that, I'm going to give you a kind of a description of how hybrid breeding compares to genetic engineering, and why genetic engineering is probably more useful for restoration. And again, let's just think about tree height here. Now, chestnut has around 30,000 gene pairs. That means 30,000 genes from the mother, 30,000 genes from the father. Okay, there's quite a few genes there. And so hybrids, about half of those will be genes from the Chinese chestnut, if you're crossing with Chinese chestnut. And the backcross breeding program, which is much better than just a hybrid, still is about 1/16 Chinese chestnut genes. Now, I like to use this illustration to kind of get you this picture in your mind of what's going on. And I like the book illustration. And let's say this book is the genome of the chestnut. And the words are the genes in that genome. So you have this book that describes a chestnut. Well, in a backcross chestnut, that's 1/16, which is pretty good. It's mostly describing an American chestnut, but there's still about 10 pages or around 1,800 words describing the Chinese chestnut. Now, why is that important? Well, one of the things you want to make sure that in these words or these genes, you have the ability to make it more resistant blight. So you're trying to retain those words. But at the same time, you're trying to get rid of these other words, such as make me shorter or make larger nuts or other characteristics that make it less adapted to our eastern forests. So it's a very complicated process. The American Chestnut Foundation has done a great job moving along, but they're not quite there yet where they have a fully resistant American chestnut tree that is also fully American traits, okay? So I want to compare this to what happens with genetic engineering. Again, using the book example, the words in the book are the genes, and we start with a book that totally describes the American chestnut. And we'll pull out a passage from the book just as an illustration here. And this passage reads, "It's very exciting at that season to roam the then boundless chestnut woods of Lincoln." This is written by Thoreau, who was a chestnut enthusiast. So with genetic engineering, basically what we're doing is making a very small change. We're only adding 1, 2, or 3 words to the whole book, okay? So now this whole book, again, describing American chestnut, you get to this passage, it reads, "It was very exciting at that season to roam the then boundless blight resistant chestnut woods of Lincoln," okay? So nothing else has changed. Everything else describes an American chestnut. So what you have is 100% American chestnut plus blight tolerance, which tolerance is a type of resistance. Okay, so how do we know what genes to add? What do we use? Well, one of the things you can do is look at, well, what causes resistance in the Chinese chestnut? But that gets to be complicated because there's actually many genes that enhance the resistance in chestnut. It's requires many genes, each one individually only ups the resistance slightly. So I like to do is to look at actually how does the fungus attack the tree and then try to mitigate those characteristics, those virulence factors. So all the way back in 1978 before I even went to college, I was an airman in the Air Force in 1978. But people were publishing how the fungus actually attacks the tree. And one of the things, like in this paper, they were talking about the oxalic acid in combination with an enzyme called polygalactoranase. Okay, so these are 2 major virulence factors, or ways that the fungus attacks a tree. So can we counter those and therefore make the tree resistant? Well, for oxalic acid, it turns out there's actually enzymes that can remove this acid, which I'll talk about in just a minute. And for the polygalactoranase, this enzyme, there are actually inhibitors made by plants that can inhibit that particular enzyme. They tend to be very specific, so you have to pick the right one. So what we have done, we started off countering the oxalic acid with the oxide oxidase, and we are now, right now, cloning the PGIPs to counteract this other enzyme. So I'm going to talk mostly about the oxalic acid. So we cloned an oxide oxidase from wheat. The reason why we chose the one from wheat only is because it was the most published of the enzymes and genes. So we want to always start with something we know the most about. But this is really ubiquitous in plants and fungi. By the way, I need to always mention this is a non-gluten enzyme since it comes from wheat. It's also a non-allergen. So if you look at this oxide oxidase, it can be found in all kinds of crop plants. You probably eat this every day. It's in peanuts, oats, sugar beet, tea, so on and so forth. It's also found out in the wild. These are some wild plants that people have discovered oxide oxidases from, and there's the references. And even in fungi and bacteria. So this is nothing new to the environment. This is an enzyme that's out there already. So what does it do? It detoxifies the oxalic acid. It takes it, it combines it with oxygen and makes hydrogen peroxide and carbon dioxide. These are 2 products a plant uses all the time. One for making lignin to help shore up the cell walls, And carbon dioxide, of course, is used to make sugars. This is not a pesticide. It's more like an antitoxin. So it's not really killing the fungus, has no cytoactivity. And since it does not have this cytoactivity, it tends to be more stable. And that's why so many plants use this defense gene. It's a very stable, and because there's not a lot of selective pressure to overcome this particular gene. That doesn't mean it doesn't help that it doesn't mean that we shouldn't look for other genes to stack with this, which we actually are. As I mentioned, we're looking at the PGIPs, but just by itself, it's going to be very stable. And with other genes, it will become even more durable for the future. So how do you get a gene in? How do you get this gene in that we cloned? Well, we use a natural genetic engineer. It's called Agrobacterium tumefaciens. This bacteria normally genetically engineers plants in the wild, and it does that so that it makes the plant make food that only that bacteria can eat. So it does that in the wild. There's actually sometimes when it transforms plants, it actually leaves genes in there. In fact, all the sweet potatoes that you might consume today are what we call natural GMOs, because they've been engineered by Agrobacterium around 8,000 years ago, maybe 6,000 years ago. And there's been recent studies. We live in an age of genomics where people are sequencing genomes all the time. And there was a study looking at a lot of the dicot or broadleaf plants, their genomes, and looking for Agrobacterium genes in there. And what they found was about 7% of the plants they tested several hundred, that about 7% actually had genes from the Agrobacterium, or at least sequences from the Agrobacterium, showing that yes, this is a natural genetic engineer, and this is going on in the wild all the time. In fact, walnut trees are one of those ones in the list of engineered plants. So that's what we use. But when we use that in the lab, it only puts a gene into one cell at a time. So you have to do something else to get the whole plant. And that is be able to regenerate a plant from a single cell. And that's where tissue culture comes in. And this is next series of slides we call how to make a tree from seed the hard way. Basically, what we had to do for chestnut anyway, is to isolate what's called somatic embryos. There's about a 2-week window after you pollinate the trees that you can get immature embryos out of the chestnuts. And as you can see, these nuts are very small at this point. And we sterilize them, we dissect them under a microscope, and those little blobs in that circle are the actual embryos. Unfortunately, only about 1 out of 1,000 will produce a somatic embryo culture. So it's a lot of work to get these things established. But once you have them established, they will keep budding off new embryos. So all those little blobs there you see in that picture is a new embryo, and they all start from single cells. So if we put the gene into a single cell, it will make an embryo, and then we can go and we can maintain that for a number of years. This is what we use for the Agrobacterium transformation. But then we can regenerate these. And we do this through a number of steps that basically mimic what goes on inside of a seed. We change the hormones, change the sugar and salt concentrations to have it go through its developmental stages. And eventually we ended up with shoots at the end of this. Dr. Maynard said each of these steps was about a master's degree project to get through them. So we end up with shoots, so we can have actually whole forests in a jar, so to speak. But the plants aren't ready to go out, of course. They have no roots, they have no cuticle in the leaves. So we have to go through a number of other steps. We have to do some rooting, we have to do acclimatization, and finally we get plants that go out in the field. And we've been testing our plants in the field under USDA permits since 2006. So how do we test for blight resistance? Well, we have several different tests, but the one that we use mostly is what's called a small stem assay. And I'm going to show you a time-lapse photography of a really small stem assay. We usually do them bigger than this, but this is a very small one, and kind of see what's going on. Got a little music to this, okay? So it's going to start in just a second. Okay, what you're looking at are, on the left is a LS1, which is the American chestnut. The King or Qing is the Chinese chestnut next to it. And then we have 2 of our transgenic events over on the right. Each have been infected with a fungus, Cryphonectria parasitica. And you see those little wraps in the center? That's where the fungus is. Now this is over about a 2-week time period. And watch what happens. seeing is that the fungus has girdled that stem, and that American chestnut is wilting. And that's what happens to American chestnut. The Chinese chestnut is surviving, and the two transgenic events, or genetically engineered ones with the OXO, are surviving also. So we've done this test over and over again with small stems and larger stems, and we're very confident that this confers a lot of blight tolerance. We call it blight tolerance because you're not actually killing the fungus, where the fungus still survives. What we're doing is just taking away its weapon so it can no longer girdle the stem. So the question is then, is this trait inheritable? Can it be inherited to the next generation? Well, so we couldn't just wait and grow trees out in the field until they pollinated, because that takes, 3 to 7 years. So we developed a method to do quick pollen development, and we use a high light growth chamber. And with this technique, we can actually get pollen from our genetically engineered lines in less than 11 months. Then we can take that pollen, outcross it to wild type American chestnut trees, and collect those nuts, and about half of them will inherit the oxalate oxidase gene. And these trees are also, that inherit the gene, will be blight tolerant, as shown in this series of assays. So we know that the tolerance is inherited. Now, that doesn't mean the trees don't get any damage. Now, I'll make that clear, because even Chinese chestnut gets damaged from the blight if you inoculate it. And you've got an example of a 1 centimeter stem of Chinese chestnut on the left. We have our oxalate oxidase tree in the middle, and the wild type on the right. So it's kind of very similar. And this was always our goal, is to be at least as resistant as the Chinese chestnut. On the right, you got a deadly canker. It's completely sunken, and the top of the tree is dead. Okay, so now we have this. Before we can get our trees out to the public, we have to go through a federal regulatory process where we get approval. But before we even submit to them, we had to go through a number of experiments to show that our trees are no different than the wild type, except for the blight tolerance. So we've looked at things like mycorrhizae, the good fungi that help trees that colonize their roots. We've looked at bee feeding on pollen. We looked at the nuts, the nutritional content, and other chemicals that are in the nuts, and there's no difference in those. We've looked at leaves, feeding them to aquatic insects and terrestrial insects. We even fed them to wood frog tadpoles. And this was kind of an interesting experiment we did. And when we did it and looked at the development rate of the wood tadpoles, we actually compared our trees to not only the wild type American chestnut, which there was no difference you see there in the center between the GE and the wild type, but we compared it to other trees too, such as Syrah maple, American beech, hybrid chestnut, and Chinese chestnut. And it turns out that the American chestnut actually did better than all those others. So getting the American chestnut back probably will have many benefits. Now that we have this gene that confers tolerance and it acts like a dominant gene, we can actually go out and start rescuing genetic diversity and local adaptations of the chestnut tree, because there's still millions of chestnut trees out there. So our original chestnut tree that we made would be a clone, and we don't want to put that out in the field as a restoration tree, because it doesn't have the genetic diversity we want. So we are in a program now of breeding, where we're outcrossing these trees to wild mother trees, and their offspring tends to be about 50% inheriting the oxalate oxidase gene. We have an easy test that we can test either leaves, or we can do nut cores to find out which ones inherit the gene. And each time you outcross this, you get different complements from each parent. And therefore you start building up diversity. We are going 1 to 3 generations for a horticulture distribution, and 3 to 5 generations for a forest restoration population of trees. So we are doing this right now. We're already up to the 3rd generation and working on the 4th. One of the things that when we finally distribute the trees, not only will we distribute trees, but we're going to be distributing pollen for people who want to actually do some pollination of their own wild type trees. One of these slides can pollinate at least 20 to 40 nuts, or birds, I should say. So if you're interested in that, we actually have this year a virtual pollination workshop that you can go and watch if you want to learn how to do that. And once we have regulatory approval, we can actually send out pollen And you can get a lot more trees that way than just getting individual trees from us. Now, one last thing I want to discuss here is that we are starting what's called common garden experiments, because one of the things when we make these trees, well, one thing you have to understand is that resistance is not a single thing. It's a gradient. Chinese chestnuts have some different levels of resistance. For example, some Chinese chestnut trees will have less damage than other chestnut trees, and the ones with less damage are more resistant. Even American chestnut trees have different levels of resistance in them, even though they all die. Some die quicker than others. So what we're doing with the addition of the oxide oxidase is actually interacting or working with the genes that are naturally in the tree. So we're pushing that tree above that line so it can survive, okay? But those genes are important. So as we outcross the trees to different genetic backgrounds, different mothers, you might have a tree that's more susceptible to start with, and therefore you only push it up so far, and you might get one that has a little more damage. Or you could have one that's more resistant and push it up even farther and have less damage. So we're actually testing this hypothesis that we're going to get a varying amount of resistance, just like the Chinese chestnut. And we're planting this coming summer common garden experiments in 4 different states to actually test this particular hypothesis. So as I mentioned before, when we do these outcrossing, you have your transgenic tree shown on the left there and a wild type tree, and you get about half of them are going to be with the oxide oxidase, and that's because you get a chromosome with the oxide oxidase gene from each parent. Okay, so about 50%, 50%. Now this is important because this is why in conservation, this is kind of unique. We could have actually trees out there that are hundreds of years old or 100 years old. And even after 100 years, we can still get the original wild type American chestnut tree back. So in the future, as techniques get better and better, we can always recoup the original chestnut tree and try new techniques with that if we wanted to. You can't do this with other breeding methods because those changes basically can't be bred out, where this one you can. Okay, so how do we do restoration? Well, we're going to be planting them in a lot of different places, but it's not going to be rely on, we can't rely on ourselves with this. This is something that the people will have to do. And we call this a century-long project because it's not going to be quick. Chestnut trees do not travel very quickly on their own, only about a mile or 2 per 100 years. So, it really is going to rely on people actually planting these trees, okay? And we hope that our work that we've done with American chestnut will act as a model for other tree species that are endangered from exotic pests. For example, this picture here is tan oaks that are being killed by sudden oak death out in California. So, genetic engineering is not a magic bullet, but it is a good tool that can be used along with other tools to possibly save other trees. And so I just want to give a shout out to my team. We typically have about 25 people on the team at any given time, but over the years, we've literally had hundreds of people working on this project, and it's all due to them that we are where we are today. And I'm just going to close with this statement from Dr. Robin Kimmerer, who talked at the UN a couple of years ago. And I like this quote, "We humans are more than consumers. We have gifts of our own to give to the earth." And that's the way I view this chestnut project. We're giving something back. It was humans that brought this disease over to the United States and started the chestnut blight. And I think it's up to us to actually stop it. So I'm going to thank you at this point, and then I'll start answering any questions. And let's see if I can stop this sharing here. Okay. Oh, I went a little over time, but close.
A lot of information in a tight time frame. Well done. Let's take a break and then we'll take some questions. And I'll just remind everybody, there's a Q&A box down below, and a couple of you have put a few things forward. If any of our listeners today would like to join us at the American Chestnut Foundation, go to www.acf.org and click on the membership tab. Members will receive our monthly newsletter, TACF's Chestnut Magazine 3 times a year, invitations to our spring growers and fall meetings, volunteer opportunities, and have access to chestnut seeds, weather and growing conditions permitting. Plus, really, you get to meet and work with people who are pursuing something larger than ourselves, and that will span many generations. This year, our chapter has thrown in an additional incentive. Any new members who join the PA New Jersey chapter in January will automatically be entered in a drawing to win 6 pints of ice cream of your choice from the beloved Penn State Berkey Creamery. Creamery is located about 2 minutes from our headquarters office at Penn State and about 10 minutes from our chestnut orchard. And when people come to visit with us, they usually find their way over there. So let's take some time now to ask some questions. I see there's a few here already. Thank you. So Bill, I think you began to address this. Are similar approaches being used for elm or ash or any of these many trees that are really under siege right now?
Yeah, there's actually been some genetic engineering of elm trees. Not elm, I'm sorry, ash trees. There was some work at Purdue for a long time. But, I think most researchers are kind of being cautious right now because the regulatory process is very daunting. And you really have to have time and finances to get through that. So I think a lot of researchers, they do some of that research. I think there's been some genetically engineered walnut, but they haven't moved forward as far as actually doing a restoration or trying to get them out for restoration because they're waiting, I think, for us to show that it can be done. But there are other people very interested in it. And once there's a clear path forward, there's probably going to be a lot more people jumping on this and trying to rescue trees. We are actually interested in ourselves. We are working with American elm. We also work with some chinkapins, but we are interested in actually going out and working with other trees once we have our chestnut going through the regulatory process.
Okay. I gotta ask, where did the term, I noticed the term, I know that the petition to the USDA is for the Darling 58 American chestnut. And I noticed on that beautiful, what do you want to call it? The graphic, the video you had.
Yes.
There was Darling, I think 212 or 230 something. And I'm curious, where did Darling, where did that come from?
Okay, so Darling actually is named after Herb Darling and his family, the Darling family, mainly because Herb was one of the original people that came and talked to Dr. Maynard and myself back in 1989, way back then. And he's actually was the first president of the New York chapter. I think he was one of the founding members. And he was supporting us for like 25 years before he retired. And we just thought that it would be nice to kind of honor him in his long-term support and undying enthusiasm for the tree. So we named it after him.
Excellent. Ray asks, any suggestions on how the general public can urge the relevant regulatory bodies to approve these chestnut trees?
Yeah, so we just recently went through a what's called a public open comment period with the USDA. That was a 60-day open comment period. We could really use help during those times. We're going to have several of them ahead of us where people would write in and for their support, either just saying that, they want to plant these trees. If they have any kind of science background, kind of giving some information about that, or environmental background. But, it's generally just supporting our trees. It's very, very helpful to have positive comments sit in during these times. The next one's going to probably be sometime this spring. And it's going to be on the environmental impact statement that we're not writing. Actually, the USDA is writing using the information that we gave them as well as their own research. And that should be coming out hopefully sometime this spring. So keep track either at the TACF or ESF for when we will request you to write in at those times.
Yeah, and I would just add that's a benefit to being wired to what TACF and what ESF is doing, because both organizations go out of their way to make their mailing lists and help people understand their role and the importance of public comment. Living in a democracy, the public really does have an impact. So, and we want to thank actually thousands of people who wrote
Yes.
in. Yes, the scientific community wrote in, but also many members of the general public voiced their support for this tree to USDA in the recent public comment period. And there'll be more, as Bill points out, there'll be more.
Right.
John asks, do we know very much about why certain introduced insects are attached, I think, to a specific species of tree? So for instance, an emerald ash borer attached to an ash species.
Yeah, basically they usually come from a part of the world where there's a related tree species. So they kind of co-evolved with those trees. So they have the mechanisms to attack those trees, but those trees also have mechanisms to defend themselves. And there's always this balance between pathogens or pests and the host, the tree. And, you get kind of tit for tat as things go along and all. But when you move a insect or a fungal pest or whatever from one part of the world to the other, all of a sudden you're getting to related trees. Again, for example, the Chinese chestnut and American chestnut are very closely related. But the American chestnut had never been exposed to the fungus. And same thing with the ash trees here, have never been exposed to that particular insect. Yet that insect has all the necessary equipment to attack an ash tree. Okay, so that's the problem. The problem is moving these pests around the world and into, we call them naive populations of trees that never been exposed to it, and so they don't have the mechanisms to defend themselves. But the reason why they attack a certain tree is because that's basically what they attack in their last ecosystem.
Let's see. Okay.
Yeah.
Got a question. In Pennsylvania, particularly, spotted lanternfly is all over the place in southeastern Pennsylvania, and probably coming to a theater near you, unfortunately. Is there any evidence, or would you know of any evidence that would suggest spotted lanternflies like chestnut trees?
I haven't seen that yet, but you never know, because it's kind of has a broad host range. So there's several different trees. I mean, there's certain ones that prefers, and there's one, what's it called? Is it tree of heaven or something? Like that. There's certain trees that it prefers, but then there's other trees that will also attack.
Right.
So we just don't know yet. I think you guys in Pennsylvania might learn first if it does, if you start seeing it on chestnut trees. It's not, at least as far as I know, it's not here in New York yet.
Well, it sure likes maple. It sure likes red maple for some reason.
Oh, okay.
Yeah, it's kind of frightening, which and it's just it's a long trail of, as we move plant material around the world. We're probably exporting our invasive our pests overseas
Yep.
and having a similar impact in various
Oh,
places.
absolutely. It's funny because it's probably been 15 years now, but I had an opportunity to go to China to look at chestnut trees there in China. And when we talked to some of the people there, they said, oh yeah, we have the same problem with invasive species coming here, again, because it's when you move a species from one ecosystem to another that's naive to that species or that pest, you have the problem. And these parts of the world are separated by huge oceans, so
Yes,
normally
yes.
travel, but they can travel on an airplane or they can travel on a ship. So
Yeah, it's amazing. So Michael asks, does the 50% passing on to offspring of the blight the 50% passing on to offspring of the blight tolerance hold at 50% for continuing generations?
okay, so it will, as long as so the tree is in what's called a hemizygous condition, meaning that there's pairs of every chromosome and it's only on one of those pairs. So in that condition, it's always going to be 50%. And I must mention too that you don't always get 50%, because when you're out there pollinating, sometimes you get wild type pollen coming in and pollinating your plant that you think you pollinated with your transgenic pollen. So sometimes it's, maybe 45% or something like that. But it's close to 50%. So that's going to always be that way until you start intercrossing those trees. Okay. When you intercross those trees, then you get, 25% will be homozygous, 50% will be hemizygous, and 25% will be wild type. And then if you cross those homozygous ones, meaning they have both copies in both chromosomes of the oxalate oxidase, when that crosses out, you'll get 100% offspring. Okay. But if you think of it as a population, there's always going to be wild type being produced. Okay. Because you're never going to get 100% homozygous. You're always going to have some kind of mix. But right now, we are just actually trying just to do the hemizygous outcrossing, where we only have one copy in every tree, just because it allows us to increase diversity faster. We're not worried about going to be intercrossing these and making homozygous yet.
Okay. This may be a similar question, but here's Robert asks, it seems that there is some partial confidence that resistance will persist at least 3 generations, but how do we know it will, how do we know if it will gradually reduce through successive generations?
Okay, so unlike when you're working with quantitative traits and do breeding, oftentimes the more outcrossing you go, the less resistance you have, because you're actually segregating out those genes that combine together to make resistance. This is a single gene, so that's not going to happen with this particular one. Now, as far as stability of the gene, that's going to be the same as any other gene, whether it's from a breeding program or whatever. We'll see how long it stands up, but it's gone through 3 generations. It's been out in the field since 2006, and we don't see any problems with durability with that yet. So, but I can't stay here and say 50 years from now something might change, but it would be the same for any way that you develop these trees, whether it's a hybrid or anything. You have that same problem.
Okay. Bill, that again, going back to that little video, it was quite dramatic and a little bit sad to watch the American chestnut just wilt away in a 2-week period of time after being inoculated with the blight. Mm-hmm. Many of us who have planted trees have seen that play out over a year or so. Many of us who have seen trees that are American chestnuts that have sprouted from their stumps, we'll get to like 10 inches and then get attacked by the blight, and a year later they're gone. Mm-hmm. What's the logic for those of us steadfast people who will still plant pure American or wild type Americans and watch them grow? What's the benefit?
I encourage people to plant wild type American chestnut trees. And there's a couple of reasons. One is to just have a conservation planting, just to keep up the genetic diversity out there, even if it's going to die in a few years. We planted some in our Tully land. Actually it's that hybrid, but anyway, and it wasn't for 9 years. 9 years they looked perfect. They didn't get any blight. But then once the blight came in, about 3 years, it kind of really done them in. But during that time, you can keep those trees alive, and you can actually get nuts from those trees. Okay, and then you can increase the genetic diversity by keep breeding these trees. They don't breed very well on their own in the wild because there's usually not 2 mature trees close enough together.
Yes,
they can actually cross. And you remember, it takes 2. They both have male and female flowers, but they're self-incompatible. So you have to have 2 different trees to cross. So I would definitely, and then here's the other thing, as I mentioned in the talk, we actually hope to be a distributor of pollen. And we've developed these actual methods where we can put the pollen on the slide. We can store it like a year easily in freezers, and then we can just mail it out to people, and they can do crosses with their own trees. So instead of asking us, oh, can we have a tree, and you go and get 1 tree or something like that, you can give you a slide of pollen, and you might get 30 trees out of it.
Wow.
So the pollen, is a much better way, and at the same time, you're helping us increase diversity by crossing to your trees in the local area.
Mm-hmm. Okay. Excellent. Another question, how many different darling varieties have been developed and how many are you hoping to release for restoration?
Well, I mean, that's a hard question to answer because we weed out things from the beginning. I mean, you start with basically, when you do a transformation, you get hundreds of little spots of transformed tissue, and then you grow them and you're weeding out all the time which ones will get develop into shoots, and then you have so many shoots, and then you do things like, okay, how many inserts of the gene went in? We only want one, so we weed out the ones that have multiple inserts. How much is the gene being expressed? Because it depends on where it goes in the genome will help control how much of the enzymes being made. And so we do expression studies and we weed out all the ones we don't want from there. And then, you keep going and going and going. And then you finally, do your tests and all. And we end up with one. Only reason why we chose one to go with the regulatory is to simplify things. It's much easier for them to review one plant than a bunch of them. And in fact, that's why we start with just one gene also, because the regulatory system is so complicated. It's much easier just to go with one gene than multiple genes. Okay. So yeah, so it's hard to know. It is hundreds though.
Okay. Do you have a feel for the USDA approval timeline? And have you gotten, Edward's asking this question, have you gotten any early comments from USDA?
Well, they have talked with, we've been talking with them as well as the EPA and the FDA for 5 years now and they all said this is possible, and basically been very helpful. They have to follow all the rules and all that kind of stuff, but they've been very helpful to teach us how to actually apply and get this stuff reviewed. So I think, we have an excellent chance of being approved because we've done everything that they have said for us to do. Now it's just a matter of going through and having it reviewed. And like I said, it was a 290-page document to the USDA. We're almost having all the documents, which are completely different kinds of documents, that go to the EPA. And their timeline is 20 months for their review. The FDA is actually the simplest one because they leave all the environmental stuff to the other agencies. And they're mostly interested in nutrition, making sure you don't have any allergens, making sure that you don't have any toxins in there, which we don't have any of those things. So, and the nutrition is normal. So we've done all those tests, and we'll be submitting to them last just because they are the shortest and easiest of the three.
Okay. Hey, is it true that chestnuts are gluten-free?
Yes, it is. Yep, they are gluten-free. That's why I had that one picture of a beer, because people can actually brew beer out of chestnut, and it's a gluten-free beer. So they have no gluten in them, and just because we use a gene from wheat, they still have no gluten in them because we're not using a gluten gene.
Okay. Well, I do know chestnut liqueur is a big deal over in Europe. So.
Well, they do all kinds of things with chestnut. I mean, they got, chestnut candies and, and flour for baking and, all kinds of different things.
Yes.
They've been using it for a long time, ever since Romans,
Yeah, in many ways, it's almost like Asia, where chestnuts are revered and, are a fall tradition, and Europe, similar situation. They have festivals. It feels like those cultures are using chestnut in full living color. Mm-hmm. And so many of the pictures that we look at here in the US, North America, are in black and white. And it's almost right. Like at one time it was all in living color. It was all part of the joy of living in the Eastern United States.
Yeah, and there's so many uses. I mean, I've read papers about people looking at using chestnut as a feed for hogs. Yes. And actually improves the flavor of the and consistency of the pork. And it brings me back to United States when chestnuts were around. The subsistence farmers used to release their hogs into the forest in the fall to get fattened up by eating the chestnuts, and they probably had some good eating there.
Okay. Bill, what's ahead? Technology moves forward, and I think of it in partly in context of, we're all kind of locked we're not totally locked down, but we're pretty well locked down. On the other side, technology keeps moving forward
Mhm.
through recessions, prosperity, pandemics, et cetera. And just as everybody accelerated things to try to come up with a vaccine, there's just so many fronts that the technology moves forward. Rich asks, would CRISPR make the gene transfer in the embryos we develop easier?
Yes. The answer is yes, but, and we are working on that right now. Unfortunately, when you read these articles about CRISPR and how easy it is, it's not quite as easy as you think. Most of those things were developed on what you call model systems that people know really well. For trees, there's a lot of hurdles you have to get over, which we are working on right now so that we can start using CRISPR. And the advantage of CRISPR is that you can go in and make even smaller changes than what we made.
Right.
You can make a change to an actual gene that's already there, maybe making it so it makes more of that particular enzyme or something like that, or changing in a slight way. So it's actually less of a change than even the old-fashioned genetic engineering methods. So we are working on that. I have a couple graduate students doing that. But before you do it, just like when we did the transformations, it took us many years to develop the techniques to do it. And so we are doing that right now. We're developing those techniques to start doing that. And we hope to actually use it not only for chestnut, but for other tree species.
Did you mentioned small stem assays? I'm just curious, did that come out of your program? Was that?
Yeah, pretty much. I think other people might have tried it, but they never really reported it. We actually published in the Chestnut magazine a number of years ago our methods. And again, we wanted to have something that we didn't have to wait, 2 or 3 years for something to grow in the field before we can inoculate it.
Right.
And so we did a lot of tests and found out that, well, the small stem assays can give you a good indication of resistance. Matter of fact, it's actually such a severe assay that a lot of times, you can't really measure small increases of resistance. You can only measure larger increases of resistance because even Chinese chestnut, if you inoculate it, sometimes those small stems, it will girdle it and kill the tree, So, yeah. So
Okay.
yeah, we kind of developed that too. Again, a lot of what we do is developing techniques. I mean, we
Right.
developed the technique to get the early flowering. We developed those small stem assays. We developed a leaf assay that actually gives us a preliminary test before the small stem assays. We had to do all that kind of stuff. We had to develop all the tissue culture techniques with some help from other people like Scott Merkel at the University of Georgia. He was very instrumental in the tissue culture. But a lot of things were kind of, I used to always say we have to build a boat before we go fishing. That's what we do a lot.
Yeah, it's like one of these really huge benefits that comes off of all this work. Other scientific advances that can be applied elsewhere. This is kind of a technical question, at least for me as a non-scientist, but will incompatibility be an issue if and when the transgenic variety is released? And will they all be clones?
No, they will not all be clones. And actually the self-incompatibility helps with that because they can't self, they have to outcross to another tree, which will help increase diversity. But even before we actually releasing those trees out there, we're not releasing the original clone because that's how you have to make it first. You make a clone. But then we've been outcrossing already 3 generations to a lot of different, and we're doing pedigrees and following the trees, make sure we are trying to get local adaptations. So southern trees to southern trees, northern trees to northern trees, and such as that, and as much diversity as we can in there. And we're going to have a couple releases. One would be, again, a horticultural release, which we are looking at. These are going to be smaller plantings of 20 trees or less, probably, where we have enough diversity for those kind of plantings. But then when we want to get out to the thousands of trees, we really want to have a more diverse population, and that's what we're working on right now. When we're doing this in collaboration with the American Chestnut Foundation.
Okay, excellent. Well, we've about reached the end of today's session. And I think one of the things I just take away from this is just the importance of preserving, the importance of growing wild type American chestnuts because when we have them and it's going to take several years for lead time on all this good stuff. These could all be part of the process of diversifying the population and being able to move forward. So we appreciate all the work that goes on with
Yeah, and let me just add to that. If you want to plant wild type trees, and I really encourage you to do that, try to get ones in the local area. Because again, we want to increase that diversity. We don't want to have trees all from one location being crossed with our trees. We want to have a very diverse population. So.
Well, thank you, Bill, for your inspiration, dedication to finding a way to develop an ecologically sustainable blight tolerant tree, and for giving us all hope and optimism that, one day we will be able to save other trees that are under severe threat from the new pests and pathogens being introduced around the world. Thank you also to all our members and all of you who joined today. Join us all this week for a new discussion about all things chestnut. Tomorrow on Thursday at noon, Sarah Fitzsimmons will discuss growing, cultivating, and restoring the American chestnut. Followed at 7 p.m. tomorrow, I'll be hosting Chestnut Growers of America's president, Roger Blackwell. Be looking at growth opportunities for chestnut in our area. And then we'll wrap up on Friday with Lake Grabowski talking about finding chestnuts in the wild. On behalf of the Pennsylvania and New Jersey chapter of the American Chestnut Foundation, we appreciate all of your interest, support, and questions. Very good questions today. Thank you, audience. More information is available at www.panjtacf.org. And thank you. And we hope you all will consider joining with us to help save and restore the American chestnut. Take care.
Thank you.